Hello, this is Cherry Lu from Department of Life Science in National Central University. I'm very great honor to present our latest bioremediation solution for organic halide groundwater contamination with Dechlorination biogel. So we are focused on a contamination we call the coral essence. This is a worldwide use for the clean oil and dirt in many fields, such like industrial and laundry. So there has many kinds of coral essence, such like trichloral essence, dichloral essence, and amino chloride. So when the trichloral essence TC they leak to the ground, they will go to the groundwater labels due to the density is higher than water, and then it will make the plant in the groundwater label due to its absorption by the soil. So this pollution plant is very hard to remove. In the groundwater, there's only the microorganisms that can degrade the trichloral essence. So the microorganisms, they will remove the chloride from the coral essence step by step. But when this process, they will always produce the amino chloride, which is more toxic, and then it's a human carcinogen. So the mediation will be stopped and no longer to dechlorination in this step. So this is a very big issue to remove the amino chloride. So right now it's the only one bacteria we call the Dechlorination macatis. There is only one strain that can completely degrade the amino chloride to the non-toxic compound ethene. So in our teams, we are looking forward these functional bacteria. So we collect many groundwater samples in Taiwan and then culture the anaerobic consortia. Our team has the ability to isolate the native Dechlorination macati. We call the DHC in Taiwan. So here is a DHC strain single colony. So we can isolate the very pure DHC, and then we can test its dechlorination activity. And we can use in the molecular biology tools to identify whether it is normal strain. So here is our strain DHC, we call the CWB2 in our labs that has a very unique special ability. They can degrade the TC into the essence without producing a second contamination product, such like amino chloride, BC, and then dichloral essence. So this strain, they can rapidly degrade the total chloride without any BC accumulation, so we call this is directly dechlorination of TCE. However, the other strain, they were stepwise dechlorination TCE with accumulation BC. So this is a big process by using this strain. So based on our DHC strain, we have a technology commercialization, so we designed some product for the real-world application, such like a bioaugmentation culture and then the immobilized biogel. This is our bioaugmentation culture we call the CW+. It's containing the DHC, cell number is more than 10 power 11 per liter, and then we will put in a specialized anaerobic tank so it can produce the bacteria. We can produce 300 liters output per month in our labs. And then we already applied 1,000 liters in the Taiwan contamination site. How do we use this bioaugmentation culture? So here has a factory that did the coral ethene pollution in the groundwater system. So here has a high concentration. We call the contamination hotspot. So we will drill the well into the groundwater label, and then put our bioaugmentation culture into the groundwater, and then they will accelerate the dechlorination. So here is our bioremediation achievement. This is a site with the TC contaminate. So the TC concentration is over the legal limitations. It's a very huge area. After 1 month, we inject our bioaugmentation culture. You can see there has rapidly decreased the TC concentration, and then they were lower than the legal limitation. here is our another product we call the immobilized biogel. They will we will combine the bacteria inside the silica gels, such like the hydrogen-producing bacteria we call Clostridium butyricum. They can produce the hydrogen to the dihalococois macati. So the DHC can degrade the TCE and essence by very quick. So we already tested this gel can make the long-term use in half years, and then we can put the gels into the column, so we can setting into the groundwater level here. So we can see we can hanging in the well, and then can make a multi-depth setting. So the column will directly touch the pollution plant, so they can very quick to degrade the contamination. So we always setting our gel into the edge of the contamination site to avoid the contamination area to increase. So we are putting our gel into the well, and then they will do the long-term dechlorination in the groundwater system. So here has some picture for using our products into the real contamination site. So here we are preparing the nitrogen cylinder and some chemical we call the electron donor to stimulate the groundwater bioorganisms, and then our bioorganism culture here. So first we will prepare the anaerobic water. They can make the oxidative reductive potential is lower than -100 millivoltage, which we call anaerobic, and then the pH is neutral. So this water, they can protecting the bacteria from the oxygen, and then they can transfer the bacteria more far in the groundwater system. So we will mix our bioaugmentation culture with our anaerobic water with nitrogen, and then we will inject this mixed culture into the groundwater immediately. So that's the one is our biogel column. We will setting our biogel column in the well by hanging with the rope, and then they were ready for the long-term dechlorinations. So here is our products, and thank you for your attention to my presentation. And if you have interest in our technology, please contact us. Thank you.